Okay, so thank you so much for inviting me today to talk a little bit about my research. And I'm really glad to be here and speak for this event. I think it's very, very important to spread as much as we can the results and the new things that we're doing in the lab as a researcher to help green patients. So my name is Simone Chiola, and I'm a postdoctoral research associate at the University of Utah. And my lab is in this beautiful state of Utah, where you can see we're surrounded by gorgeous mountains. And I work in the Shcheglovitov lab. In the Shcheglovitov lab, we are mainly interested in studying neurodevelopmental disorders, especially the neurodevelopmental disorders associated with rare genetic variants. And as you can see from this picture, you can see in this dish are growing small circles of tissue called brain organoids. And in fact, what we do in the lab is start from the patients. We start from the patients, individual with green to be rare mutation in my case specifically, and we derived induced pluripotent stem cells. These induced pluripotent stem cells can be grown in organoids and further can be analyzed. We can use these organoids and do several assays to see which type of modifications the mutations induce in the development of the organoids. And in particular, we can conduct molecular genomic analysis, but we can also do functional analysis like electrophysiology. Right now in the lab, I have a project based on green to be mutations and organoids derived from patients with green to be rare variants. In particular, as future work, we want to expand the amount of green to be mutations that we're studying in the lab, and we are acquiring new lines from the SFARI Foundation. And one of the main goals is also to use brain organoids derived for these patients to test possible gene therapies that can be used to treat green to be patients. Thank you.